Thank you very much. As Sarah told you, I will tell you a few words about Epi-CRISPR targeted DNA methylation of ARCS gene as a potential therapeutic strategy in diabetes. And I would like to thank you very much for this opportunity to share my results, and also a part of the results that I obtained during my STSM in Alba synchrotron in Spain exactly 1 year ago. So as Sarah told you, I'm working in the Institute for Biological Research, and this is my group, Molecular Biology Lab. We are dealing with problems to prevent and cure diabetes. And what is a general belief is that a cure of diabetes is actually lying in the increase of beta cell mass. So you can increase the beta cell mass in two ways. By stopping beta cells of dying, or prevent them to die, or by reprogramming other cells to become insulin-producing cells. So we are doing a little bit of both, but today I will tell you more about our attempts to reprogram the pancreatic alpha cells into pancreatic beta cells. This was done in 2018 in Cell. It was a paper published by a German group that they actually overexpress several transcription factors in pancreatic alpha cells, and this turns them into insulin producing cells. We decided to do another strategy. We are using Epi-CRISPR tool for straightforward one-step pancreatic alpha to insulin-producing cell transdifferentiation by targeted DNA methylation of genes that are essential for maintaining pancreatic cell identity. In this case, it's an ARCS gene that is highly expressed in alpha cells and completely repressed in beta cells. First of all, we had two different mouse pancreatic alpha and pancreatic beta cell lines, and we performed the RNA-seq. We obtained data, and regarding the data, as you can see, pancreatic beta cell lines and alpha cell lines have completely different transcription profile, of course. So among beta cell lines, we choose NIT1 cells, mouse cell line, which produce most of the insulin 2 gene, which is responsible for glucose uptake in mice. And from data we obtained between the two alpha cell lines, we choose alpha TC16 cell line, since this cell line produce much more glucagon, and actually ARCS gene is fully active. So we performed cheap experiments, and we show that ARCS promoter in this cell line is fully occupied by RNA pol2. It has an active epigenetic mark, so H3K4 trimethylation is present, and H9 trimethylation is less present than compared to the beta cells. So ARCS gene is expressed. There is a protein synthesized. We actually show this by immunofluorescence staining in alpha cells, and these are beta cells with no ARCS. So by doing a high resolution melting analysis, we here is the ARCS gene. So the CpG island is located partially in the promoter and overlapping the exon 1 and first intron of the ARCS gene. So we analyze this amplicon in red here in the promoter region, overlapping partially the CpG island, and this amplicon in the exon part in alpha and beta cells. And as you can see, in alpha cells, there is no methylation, while in beta cells, these regions are highly methylated. So we had a perfect two cell lines that we can use for transdifferentiation, a perfect alpha cell line actually, and beta cell line where the ARCS gene is fully active. We used the Epi-CRISPR construct that was designed by Professor Tomasz Jurkowski and his team. He's our collaborator in, when this work was performed, he was in Stuttgart, now he's in Cardiff University. So Epi-CRISPR actually consists of the deactivated Cas9, and as an effector domain, we had 4 different effector domains fused to dCas9. One effector domain is DNMT3A3L, so DNA methyltransferases. Then other construct had a Cas9 3A3L fused to CRAB, and then dCas9 CRAB fused to 3A3L DNMT, and just dCas9 with CRAB repressor. And the other construct was bearing the gRNAs for the ARCS gene. We designed 4 gRNAs sitting in this position. And the first preliminary experiment was to transfect the pancreatic alpha cell line with OR4 construct together with all gRNAs or just with the gRNA 275. High-seq data actually showed us that the best working effector domain is a 3A3L fused to a CRAB. It makes the most of methylation. This methylation pattern is presenting this amplicon here in the promoter region. So all other experiments were done with pancreatic alpha cell lines transfected with this construct bearing the 3A3L CRAB as an effector domain. So our transfection was done on 5th, 7th, 10th. So we transfect cells and sort them on 5th, 7th, 10th, 12th, and 15th post-transfection day. For the transfection efficiency, we transfected also a vector bearing the GFP protein. So before sorting, we had, these are a mock transfection, and these are alpha cell transfected with the construct bearing 3A3L CRAB effector domain. So we had almost 80% of transfected and viable cells, and after sorting, it reached 90%. So from these sorted cells on different days, here I show you the results for 5th and 7th post-transfection day. So we did the HRM analysis for this amplicon here in the promoter region and in the gene body. And in both cases, using our construct, we were able to methylate these parts of the ARCS gene. And if you compare with the beta cells, the level of methylation is almost the same. It's a bit higher in our transfected cells, but without any statistical significance. But at least it's the methylation level is the same as in beta cells. So following the ARC expression, the ARC's gene expression was declined, and insulin started to be synthesized. The peak of, so ARC starts to be repressed from 5th day and 7th day, and then starts to lose the methylation, while insulin starts to increase, and the 12th post-transfection day we have a peak of insulin and mRNA, and also this was detected by immunofluorescence. So the orange is insulin in our transfected alpha cells, 3rd, 5th, 7th, 10th, and 15th post-transfection day. So in this moment, we have these cells in our hands, and they synthesize insulin, they make them in the vesicles, and also they secrete them in out of the cells upon assimilation of glucose. But the problem is that the number of cells are really low of insulin producing cells if you compare to a number of transfected cells, percentage of transfected cells, and the amount of insulin that is secreted out in the media is really low in nanograms. So we should work on these are, of course, transient transfections, so the next step would be the stable transfection, but in general, our hypothesis is working. So if you methylate one gene, and it is an ARX gene in pancreatic alpha cells, you are able to reprogram them into insulin-producing cells. So with these cells in my hand, I started STSM, as I told you 1 year ago, in Alba synchrotron in Spain. And this was collaboration with Dr. Tanja Ducic. So first of all, I brought these transfected alpha cells together with beta and alpha cells on cover slips. And we sent the cells from Spain to USA. Because sending the cover slips with the cells to USA from Serbia, it's really mission impossible. So we send the cells to Argonne National Laboratory in Chicago in USA, and Dr. Si Chen performed all the experiments. And the experiment was to use X-ray fluorescent microscopy and to trace different metal and non-metal ions within the cells. So these are pancreatic alpha cells seated on the cover slips. And we trace the phosphorus, sulfur, chlor, potassium, calcium, ferroions, copper, and zinc. And what is for us really important, it's calcium and zinc ions, because zinc is really necessary to fold the insulin in a proper manner. And calcium is ion that is responsible for assembly of insulin, its proper storage and secretion of insulin into out of the cells. So merging these two elements, you should see if the insulin is properly stored in vesicles within the cytoplasm of the cells. And we compared alpha cells. So the merging should be in white, colocalization of calcium and zinc. This is pancreatic alpha cells that should not have the insulin vesicles. These are pancreatic beta cells with merged in white calcium and zinc and vesicles within the cytoplasm. And these are the, so just one slot I show you, there is hundred of images that should be analyzed. So these are Epi-CRISPR induced insulin producing cells. And what is really interesting, it's a way how the calcium is distributed within the cells. So it is very well spotted within the cytoplasm of the cells, and it's colocalized with the zinc. So this needs to be quantified. This is just a qualitative analysis. And what is really interesting for me, it's the way how the ferroion is distributed within the cells, because there is a clear difference. Here is in transfected cells, here is in beta cells, almost no ferrion, and this is in original pancreatic alpha cells. So this is just a preliminary results. So the main part of my STSM in Spain, in Alba synchrotron facility, was to detect the DNA methylation profile changes using FTIR spectroscopy. And for this, we made, this is a job done by my master student, Ana Saric. So we made halo preparations and spin them on microscopic slides. So what are the halos actually? We first spin complete cells on the microscopic slides, and then we extracted the cytoplasm and got the nucleus only. So from the nucleus, using a high salt buffer, we extracted all the histones and all the soluble proteins. So what is left over is insoluble portion of the proteins, which used to be called the nuclear matrix. It's actually mid of last century, it was really hype. This was a structure of the proteins that actually was responsible, it was a belief in that time, was responsible for higher order chromatin organization. But what's left over from this hypothesis is the fact that part of the DNA that is called scaffold matrix attachment region is actually able to interact with the portion of non-soluble proteins within the nuclei. And this allows DNA to loop out from the nuclei. And this is called actually a halo. So we had a halo on our microscopic slides. And then we use a protein K to digest all the leftover proteins. So we obtain at the end just DNA in looped out form. So we call it a halo DNA. So how it really look like? So it's a DAPI staining. These are pancreatic beta cells from rats. These are mouse fibroblasts and NH33 cells. And these are human, I think, conjunctival human cells. So this looped out DNA actually formed this kind of halo structure. And in yellow, you can see the staining of lamin B, a typical inner membrane, protein bound to the inner membrane of the nuclei. So we also stain the halos with different antibodies. So what I wanted to show you, it's a distribution of methylated cytosine and hydroxymethylated cytosine in this halo preparation. These are mouse fibroblasts, NH33 cells. So as you can see, methyl cytosine is present in the nuclei, but it's mostly out in the looped DNA, and hydroxymethyl cytosine is more concentrated within the nucleus. So with this slides in with the halos and halo DNA, we use a synchrotron light source. So it's infrared light from 4000 to 400 wave number. And we actually spotted the spectra, recorded the spectra of each halo. So we had around 200 halos. From NIH-treated 3 cells, NIH cells treated with 5-ASA, so these cells should be demethylated, and for PARP knockout cells. These PARP knockout cells are actually mouse fibroblasts knockout for a PARP gene, so they do not have a PARP protein, and that's why they have a higher activity of the TET proteins, because we show that PARP inhibits TET. So when there is no PARP, TET proteins are more active, and they induce the hydroxymethylation. So these cells should have less methylation than proper NIH-treated 3 cells. So we compare all 3 different cell types, and these are the recordings for spectra of the full halos with the present proteins. These peak on 300, and these 2 peaks, they actually represent the vibration of the these bonds within the protein structure. This part of the spectra from 1500 wavelength to around 800 is devoted to the DNA. And these are the spectra of halo DNA, so pure DNA, from the all three cell types. And as you can see, there is no protein vibration from the bonds that correspond to the proteins. There is just these vibrations in the DNA range. And after statistical analysis using the principal component analysis, we actually have a clear difference between NIH-treated three cell lines and PARP knockout cells. In blue, you can see the NIH cells treated with 5-ASA. And since 5-ASA demethylates randomly, you will have a portion of cells that is not methylated, that is not demethylated, portion that is demethylated, and in some status in between. So that's why it's overlapping the both sides of the PCA. This is a PCA analysis in the region of 3,600 to 2,600. And the principal component analysis differences is 98%. So the major peaks that contributes to this difference is 3345 wavelength, which corresponds to the NH vibration, and 2900, which is actually symmetric vibration of the CH group within the methyl group. And NIH cells possess more of this vibration, which means that possess more methylated group than PARP knockout cells. The other part of the spectra, so this range represents actually the DNA fingerprint. And also we have a separation of NIH-treated three cells and PARP knockout cells in green. What corresponds to this difference is these two peaks. Both are peaks that correspond to the vibration of the bonds in unmethylated cytosine. So this C4, C5 vibration in unmethylated cytosine, then C2 and C4, this vibration part of the cytosine, then CO and NC in any direction. So much more vibration was detected in unmethylated cytosine, and it's present more in the PARP minus cells that should be less methylated than NIH-treated three cells. And finally, the part of the spectra which corresponds to the cytosine vibration. We also have a clear difference between the PARP knockout cells and NIH-treated tree cells. And this vibration on the wavelength of 1155 is really very important, because it's a vibration of the CH bond, C5H bond. So when this vibrates, that means that this C5 in cytosine is not bound to the methyl group. And this actually vibration is detected more in PARP knockout cells than in NIH-treated tree cells. The same is true for 1110 wavelength. It's in unmethylated C, and this is this portion of the cytosine, vibration of this portion of the cytosine. So from this preliminary analysis, we can conclude that FTIR spectroscopy can be used to detect the difference in methylation profile of DNA molecules from different cell types, different specimens. And we sorted out several wavelength numbers and several bonds in different type of molecules, in methyl group, in unmethylated cytosine, which can be used as a reference for further analysis of different DNA spectra from different cells or tissues. This would be the end, and I would like to thank my wonderful team, worldwide collaborators, of course, COST, and thank you very much for your attention. So I am open for questions.
In your first part of the presentation, so you told us that the efficiency was, I mean, it was working, but the efficiency was quite small. So what do you think is limiting here? Is it the fact that the methylation is not working very well in for all the cells, or it's that methylation is not basically regulating very tightly the production of insulins, or so? What do you think is the limiting step here?
Actually, quite the opposite. I think when the CRISPR tool is inside the cells and it's proper folded and starts to function, it's functional. It methylates ARCS promoter, and we have reduced ARCS protein synthesis, and that turns cells into insulin producing cells. But the problem is transfection efficiency is really high, but it doesn't mean that every CRISPR construct will be functional within the transfected cells. So that's the problem. We should, I don't know how to increase actually this type of efficiency, but maybe this is a transient transfection. So if we induce if we succeed in stable transfection, then I think the percentage of the functional cells will be higher.
Okay, and may I ask a second question? So, did you see that any? I mean, because you were looking at the 3D. I mean, somehow some 3D organization of some compartments in the nucleus. So, do you think that your methylation is impacting the organization that you were looking at, maybe more in the second part of your talk? And how is it affecting the disorganization?
Yeah, it's affecting, as you can see, the effector domain that is most functional, it's deactivated Cas9 foods to 383L with the crab. So of course you need additional repressor to close the complete part of the promoter. So methylation itself plus repression of the chromatin state is a winning combination. We will try to show it finally.
Very ignorant question, but it was the first time that I heard about using FTIR spectroscopy to, quantify changes in methylation, DNA methylation. I was just wondering, what are the advantages of this technique compared to, other genomics techniques to profile methylation or quantify it?
If you have a FTR spectroscopy, the machine incomplete, then you can use it as well. I mean, you can see you can compare the fine differences because it's a very strong, very sensitive, and with a high resolution technique. Of course, you have a ELISA test. You can do it. You can do bisulfite sequencing. You can do whatever. But for us in Serbia, it's pretty expensive. So we are trying the way to handle this type of a more financial problem.
If I might, very nice talk, thank you. I'm wondering, in your fusion protein for the first part you used to recruit via dead Cas9 to the promoters, you have DNMTs, you have CRAB, all these proteins interact with other proteins. I'm wondering, did you observe any side effects that any other regions in the genome are targeted by DNA methylation? Did you see this?
Do you mean off targets?
Yes.
Yeah.
Besides the site that you target by the Cas9.
Yeah, it's analyzing right now. So it's a collaboration with Tomac, as I told you. So this is a part that Tomac is doing right now. So I have no answer for this. For sure, there is off targets, but it will come.
Okay, we have a question from Ekaterina Presa. So, can you tell us more in detail how you receive these nucleoids?
So you are spinning the cells on microscopic slides, and then you are doing extraction from the slides. I mean, the cells are spin down on the slides. You are using lysis buffer, then you are using different high salt buffer to get rid of the histones and other non-soluble proteins. And also you're using Triton, non-ionic detergent, to take off the nuclear membrane. So you get rid of the lipids. So what is left over, it's this non-soluble protein mesh that is in contact with the DNA, and DNA looped out somehow. So it's complete procedure. If you want, I can send you. Yeah, high salt. Yeah, it was 2 molar sodium chloride.